So we have talked about cytotoxic T cells, how they function. Now let's talk about helper T cells. Helper T cells are crucial for defenses against both extracellular and intracellular pathogens. Basically, helper T cells orchestrate the adaptive immune response. They basically activate other cells, whether they're B cells or cytotoxic T cells, and make them and arm them basically to defend the host body. Once a helper T cell is activated by an antigen presenting cell to become an effector cell, it can help activate other cells. Here, just to remind you again, helper T cells interact with peptide fragments in association with class 2 MHC proteins, not class 1 MHC proteins, which are recognized by the T cells, which are recognized by cytotoxic T cells. Helper T cells stimulate B cells to make antibodies that inactivate or eliminate extracellular pathogens and their toxic products. So again, helper T cells are stimulating B cells to produce antibodies. Also, I would like to mention helper T cells basically instruct B cells what type of antibodies to make. Remember, we said there is a primary immune response and then there's a secondary immune response in which there's secondary class antibodies which are produced. These T cells tell B cells whether to make IgG molecules, IgE or IgA molecules. When activated by an antigen presenting cell, a naive helper T cell can differentiate into either two different types of effector helper T cells called Th1 and Th2. Th1 cells mainly help activate macrophages and cytotoxic T cells, whereas helper T cells type 2, basically Th2 cells, mainly activate B cells. And what determines what type of helper T cell will be formed, whether it will be type 1 or type 2? It is determined by basically the nature of peptide fragment that antigen presenting cell exposed the helper T cell to. So here basically you have antigen presenting cells here. Antigen presenting cells in one case are displaying their peptide fragments to T cells develop into helper T cell type 1. This antigen presenting cell is also displaying peptide fragments and it results in development of helper T cell type 2. Two cells, we have said Th2 cells activate B cells which result in production of antibodies. Th1 cells activate macrophages and cytotoxic T cells. Again, I would like to emphasize it is the nature of the antigen or invading pathogen that determines what type of response will be manifested by the adaptive immune system. APCs, antigen presenting cells, give two types of signals. Signal 1 is provided by foreign peptide bound to the MHC protein on the surface of the presenting cell, and these MHC peptide complex signals through the T cell receptor and its associated proteins. Signal 2 is provided by co-stimulatory proteins, especially B7, that is the one protein we are going to talk about. I would like to mention that B7 is also referred as CD80 or CD86. CD means basically cluster of differentiation. So same name for the same protein, which are recognized by co-receptor proteins CD28 on the surface of T cells. Too many names. I will show you a diagram and hopefully that will simplify things for you. The expression of B7 proteins on antigen presenting cell is induced by pathogens during innate response to an infection. Here, hopefully this will simplify things for you. Here is a mature APC, antigen presenting cell. It is displaying a peptide fragment in association with MHC2 molecule. And once this APC has engulfed a pathogen, it will start producing, it will mature, and that will result in production of B7 on its surface. This B7 interacts with another receptor on T cell, which is CD28. This is CD28. These two signals are required for T cell to become active. If T cell receives only, tries to become active by just one signal, that T cell will be destroyed. It will go through apoptosis because this T cell was recognizing a peptide fragment that was actually a self fragment. Since this APC is immature, it is not displaying B7. That only happens when APC has engulfed a pathogen. So this implies that basically T cells that reacting to self-antigens are destroyed because these APCs were not making the B7 molecule. So basically we have talked about how helper T cells are activated, and it is the innate immune system and also the nature of peptide fragment that determine whether the helper T cell type 1 will be made or helper T cell type 2 will be made.